Hello everyone. I'm going to talk about my project that is transcriptomic data analysis on ovarian cancer. Ovarian cancer is the fifth most lethal cancer in women, accounting for the majority of gynecological cancer deaths. In 2018, there were an estimated 295,000 ovarian cancer diagnoses and 184,000 deaths worldwide. Factors that increase your risk of ovarian cancers are getting older, being overweight, having children later or never having a full term pregnancy, taking hormone therapy after menopause, having a family history of ovarian cancer, breast cancer, colorectal cancer, and smoking. Ovarian cancer diagnosis typically involves a combination of medical history review, physical examination, imaging test, and laboratory test. Symptoms of ovarian cancer are fatigue, weight loss, discomfort in the pelvic area, changes in bowel habits, a frequent need to urinate, abdominal bloating or swelling. Treatment of ovarian cancer. The first treatment is speckle type-Os protein, that is SPOK, which suppresses proliferations and promotes apoptosis in human ovarian cancer cells by inhibiting the Hh signaling pathway. The second one is combination of Pembrolizumab, Bevacizumab, and Metronomic Cyclophosphamide. Combination of these three medicines are for the patient with recurrent platinum sensitive, platinum resistant, or refractory epithelial ovarian, fallopian tube, or primary peritoneal cancer. Now, the aim of our project was transcriptomic analysis of ovarian cancer progression and treatment. Combination of pembrolizumab with bevacizumab and metronomic cyclophosphamide response, identifying the differential expressed genes and pathways across the three time points. The study design of the treatment was based upon the novel treatment regimen. The patients which were not responding to the standard treatment were included in this study. Three drugs have shown to enhance the immune response against the cancer cells, giving pembrolizumab, bevacizumab, and cyclophosphamide together work better in the treating patients with recurrent ovarian cancer. The novel treatment regimen samples which are treated with BLBX, C4BX, and EOTBX were collected. Samples were taken from the patients at the three time points. Baseline means BLBX, after 4 cycles of treatment means C4BX, and at the end of treatment means EOTBX. So here is the methodology. The first step was data extraction. The second step was exploratory data analysis. Then we did statistical analysis, then pathway analysis, and then network analysis. So the first step was data extraction. The data was extracted from GEO database, and the title of the dataset was Integrative Multi-Omics Approach Unveils Underlying Etiology of Response to Anti-PD-1 in Combination with Anti-Angiogenic Therapy in Recurrent Ovarian Cancer. Firstly, series matrix file was downloaded from the dataset, which defines a set of related samples considered to be a part of a group, how the samples are related, and how they are ordered. Then we created the pivot chart. Here's the table showing the total number of samples of treatment. Baseline treatment had 28 samples. After 4 cycles of treatment had 21 samples, and at the end of treatment had 8 samples. And the total number of samples were 57. Then we did statistical analysis, performed PCA using unsupervised analysis. PCA is a technique used to decrease the variability between the groups and finds the outliers present in the sample. To normalize the data, downloaded raw file was run on Excel. Inside Excel, samples were grouped and the spreadsheet was transformed to text file format before uploaded to perform PCA pipeline. The tool used was PCAR library, which is a statistical process that uses an orthogonal transformation which converts a set of observations of possibly correlated variables into a set of values of linearly uncorrelated variables called principal component. Now here are the results of PCA. And the first diagram is showing outliers. After removing the outliers from the spreadsheet file, PCA was performed again, and hence the plot showing better clustering of samples. Then we did differential expression analysis. Differential expression analysis means taking the normalized read count data and performing statistical analysis to discover quantitative changes in expression levels between experimental groups. Raw data was grouped as per samples from baseline and 4 cycles of treatment. Zero errors and duplicate values were screened and removed from the spreadsheet. Spreadsheet was converted back to text format before being uploaded to perform differential expression analysis pipeline. Same criteria were used for the group baseline and at the end of treatment, and the tool used were DSEC2. DSEC2 is the popular software package used for the analysis of differential gene expression from RNA-seq data. It provides statistical methods to identify genes that are differentially expressed across different experimental conditions or groups. Enrichment analysis. These tools are software or web based platforms that facilitate the analysis of gene or protein sets for enrichment in specific biological categories or functional annotations. GSEA is a powerful tool for analyzing gene expression data and identifying biologically relevant gene sets. Now, here are the results of baseline versus 4 cycle of treatment. First one is the volcano plot, which is showing upregulated genes, that is ESM1, CYP3A4, HRG, BAAT, UGT2B4, CYP4A11, SERPing, ADH6, APOA5, and the downregulated genes are CHRM2, ENPP3, GPAM, and OXCR1. The second picture is heat map, and here are the results of heat map, in which the table 2 is showing upregulated genes in the following samples, and in the table 3, downregulated genes are showing in the following samples. Now the table 4 is showing the top 20 differential expressed gene list. The first 16 genes are upregulated and the last 4 genes are downregulated. Here's the results of differential expressed gene pipeline for baseline versus end of treatment. The first diagram is the volcano plot, and the upregulated genes are PRDM15, ACRBP, and the downregulated genes are SST, CYP3A4, APOA2, APOH, CYP3A11. Here's the heat map, and the table 6 is showing the upregulated genes in the following sample, and the table 7 is showing the downregulated genes in the following sample. The table 8 is showing the top 20 differential expressed gene list. The first 2 genes are showing upregulation and the rest are showing downregulation. Pathway analysis. After finding the upregulated and downregulated genes, we found how these genes affect the pathways of the patient. 2 major tools which were used for the pathway analysis are Enricher and David. Enricher is a web-based tool for gene overrepresentation analysis using functional annotations. David, for annotation, visualization, and integrated discovery, provides a comprehensive set of functional annotation tools for investigators to understand the biological meaning behind large lists of genes. The Enricher results for baseline versus 4 cycles of treatment. Retinol metabolism resulting in the highest differential expression value sorted on the basis of p-value ranking, and primary bile acid biosynthesis resulting in the low differential expression value. For baseline versus end of treatment, cholesterol metabolism with highest differential expression value sorted on the basis of p-value ranking and complement and coagulation cascades WPS58 resulting in the lowest differential expression value. the clustergram for baseline versus 4 cycle of treatment. In this, UGT2B4, CYP3A4 are included in the most pathways and ALDOB is included in least pathways. For baseline versus end of treatment, CYP3A4 is included in most pathways and ADH6, KNG1 and GC are included in least pathways. Now the David results for baseline versus 4 cycle of treatment. In this, retinol metabol In this, retinol metabolism, drug metabolism, metabolism for xenobiotics by cytochrome P450, bile secretion, metabolic pathways, steroid hormone biosynthesis, glycolysis, gluconeogenesis, chemical carcinogenesis. These are showing the most affected list of pathways. And for the baseline versus end of treatment, cholesterol metabolism, glycolysis or gluconeogenesis, retinol metabolism, drug metabolism, cytochrome P450, PPAR signaling pathway, metabolism of xenobiotics by cytochrome P450 are showing the most affected list of pathways. Now for the baseline versus 4 cycle of treatment, in retinol pathways, UGT2B4, ADH6, CYP34 are affected the most. And in drug metabolism, cytochrome P450 pathway, the CYP3A4 gene is affected the most. In the metabolism of xenobiotics by cytochrome P450 pathway, CYP3A4 is affected the most, and in bile secretion pathway, UGT2B4, CYP3A4 are affected the most. For baseline versus end of treatment, in cholesterol metabolism, APOA2, APOC3, and APOH genes are most affected genes. And in glycolysis or gluconeogenesis, ADH4 and ALDOB genes are the most affected. For the retinol metabolism, ADH4 and CYP3A4 are the most affected, and in drug metabolism, cytochrome P450, the ADH4 gene is the most affected. Network analysis. Gene Mania, it is a flexible, user-friendly web interface which helps in generating hypotheses about gene function and analyzing gene list. In a given query list, Gene Mania extends the list with functionally similar genes that it identifies using available genomics and proteomics data. Expression 2 kinase. Expression 2 kinase web infers upstream regulatory networks from signature of differentially expressed genes. Signature and network analysis. Signature analysis includes some sort of pattern matching of the contents of the data packets. There are patterns corresponding to known attacks. These known attacks are stored in a database and a pattern is examined against the non pattern, which defines signature analysis. Here are the Gene Mania results for baseline versus 4 cycles of treatment, in which APOA5 is co-expression, co-localization, and shared protein domain interaction with the other crucial differential expressed genes. Variations in the APOA5 gene have been associated with changes in plasma triglyceride levels, which in turn are linked to an increased risk of cardiovascular disease. For the baseline versus end of treatment, PRDM16 is showing the co-expression network interaction with SERPIND1. PRDM16 is a gene that has been implicated in various cellular processes, including cell differentiation, development, and tumorigenesis. Expression to kinase web results for the baseline versus 4 cycle of treatment. Differentially expressed transcription factors are shown on the basis from highest to lowest p-value from top to bottom of the graph, as CEPBA is the transcriptional activity with highest differential expression value. For the baseline versus end of treatment, differentially expressed transcription factors are shown on the basis from highest to lowest p-value, as HNF4A is the transcriptional activity with highest differential expression value. Protein-protein interaction expansion for baseline versus 4 cycles of treatment and for baseline versus end of treatment. ESR1 is that protein which, when affected, would lead to a cascade of reaction to affect the other proteins. Kinase enrichment analysis for baseline versus 4 cycles of treatment. In this, MAP kinases 1 show highest differential expression value. For the baseline versus end of treatment, MAP kinases 1 show highest differential expression value. Signature network analysis for baseline versus 4 cycles of treatment. In this, CYP3A4, HRG, SLC2A2, UGT2B4 are highly upregulated genes shown in red color, and CHRM2, GPAM, and OXO1 are highly downregulated genes in green color. Now, for the baseline versus end of treatment, PRDM16 are highly upregulated genes and shown in red color, and ALDOP, SST, APOC3, CYP3A4, APOA2, ITIH1, ITIH2, and GC are the highly downregulated genes and shown in green color. Then we used Venn to create Venn diagram to find the common genes. Among the upregulated and downregulated differentially expressed genes, here are the genes which were common in baseline versus 4 cycles and baseline versus end of treatment. CYP3A4, HRG, SLC2A2, GC, LDOP, and SERPing are the most common genes. Here, expression levels of genes. Figure 1 is showing the gene expression at baseline time point. Figure 2 is showing the gene expression at a time point of 4 cycles. And figure 3 is showing the gene expression at a time point of end of treatment. Here are the genes showing upregulation in 4 cycles of treatment and the genes showing downregulation in the end of treatment. Conclusion. The study found that the differentially expressed genes that are APOA5, CYP3A4, HRG, SLC2A2, GC, and LDOP were upregulated at the baseline of treatment compared to the end of treatment, and downregulated at the baseline of the treatment compared to the end of treatment. LDOP genes are detected in metabolic pathways, pentose phosphate pathways, and glycolysis gluconeogenesis. Among these differentially expressed genes, these pathways can be disrupted if the LDOP gene's differential gene expression becomes abnormally high or downregulated. The CYP3A4 gene is implicated in retinol metabolism, cytochrome P450 mediated xenobiotic metabolism, bile secretion, drug metabolism, steroid hormone synthesis, metabolic pathways, linoleic acid metabolism, and drug metabolism, cytochrome P450. These pathways can be disrupted if the CYP3A4 gene's differential gene expression becomes abnormally high or downregulated. Thank you.